So once again, these are the criteria of genetic equilibrium. So in a population that's in equilibrium, it says that the genotype and allele frequencies will stay constant over time, from generation to generation. And so the 5 criteria are shown here, and at this point I'm going to highlight different criteria and discuss specifically what impact each one has on equilibrium. So the implications of mutation. If you have a significant rate of new mutations, that can push a population out of equilibrium for a while. And the reason is that new mutations create new alleles, so they change allele frequencies. But only for a certain period of time. Eventually, as long as the source of new mutations is gone, eventually the population will return to a new equilibrium at a later time. So for example, if you think about populations of organisms that live near areas where there is a nuclear meltdown, so like in Japan or Chernobyl in the 80s. Think about rodent populations, plant populations, etc. Any kind of population nearby, initially you have a very high rate of forward mutations, new mutations in all genes. So they're showing this arrow as being pretty dark. You are going to have, at a lower rate, naturally you're going to have some reverse mutations too, but the forward mutations are going to outnumber the reverse mutations. So initially when there's a mutagen, radioactivity or some kind of chemical dumping or something along that lines, you have situation A where you have a very high rate of forward mutations. So for that time period, however long that takes, you'll have the population will not be in equilibrium. But eventually you'll reach a point where the rate of new mutations slows down. Eventually all the radioactivity will decay. And eventually, and this could be hundreds of years for some isotopes, but eventually you'll return to an equilibrium again, except it'll be sort of a new equilibrium where the frequency of alleles is changed, but from here on out, this population can reach a new equilibrium point. So the implications of significant amounts of mutation are that the population will be pushed out of equilibrium for a time, and it'll reach a new state of equilibrium later where P and Q have changed. Now what about migration? Migration, if you have migration, there can be an effect, it depends. So the implications of migration are that migration will affect the gene pool and will push a population out of equilibrium if you meet these criteria. If the number of migrating individuals is significant, so a larger number of individuals that are migrating, and if the allele frequencies of the two populations are quite different. So for example, if you have population 1 and population 2, which are genetically quite different, then if you have a reasonable sized group from population 1 that moves into population 2, you can see in this example the number of blue alleles after the migration doubles from what it was. However, if you have two populations that are very much like this population 1, if you had 2 of those, and a group from one migrated to the other, that really wouldn't change anything significant. Or if you had 2 populations that were like this population 2, if you had 2 populations like that and some individuals migrated from one to the other, that really wouldn't matter. It's when the 2 populations are genetically very different, then you can see a significant effect. So what are the effects of population size on genetic equilibrium? If the population is not very large, then it will not be in equilibrium, and it can undergo what's called genetic drift. Genetic drift is driven by random deaths and matings, and what it results in is large changes in allele frequencies in a very short period of time. And so small populations are at risk for genetic drift. So imagine you have 5 imaginary populations, and we start them all at generation 0. We start them all at a 0.5 allele frequency. So here they have allele A1 and allele A2. That's not squared. That's A2. So you have 2 alleles for a certain locus, allele A1 and allele A2. And on this y-axis, they're just measuring the percentage of A2. So let's say you start at 50% A2, which means you also have 50% A1. And then they track it over 30 generations. And because the populations are small, it's kind of this wild fluctuations up and down, because, you know, for every individual that dies, it changes the numbers significantly. But the risk is that in a short number of generations, which you can see here, here's the risk. Population 1 has reached a frequency of 1 for allele A2. That's called fixation. When an allele becomes 100% frequent in the population, that means everybody is homozygous for that allele. That's called fixation. And if you have fixation of allele A2, that means you've lost allele A1 completely. So everyone is now homozygous for A2. But in another population, you have fixation of allele A1, which means 0% of allele A2. So there's a risk of that. So this is just tracking one gene locus, but you could imagine that it's probably also happening for other loci in those same populations. So across the board, you're losing genetic diversity in the population. And then even though populations 2, 3, and 4 don't seem to reach fixation, eventually, if you extend this out another 30 generations, some of them will. So it just means that in a short amount of time, there's a high risk, not a guarantee, but a high risk that your populations will lose a fair amount of genetic diversity. And remember, the only way to generate new alleles is either mutation, new mutations create new alleles, or you could have migration of individuals from a different population that has the same species. That's also possible, as long as there are more than one population in existence. So anyway, that's the risk. Genetic drift really leads to loss of genetic variation, and that's the risk of a small population size. And even when a population is repopulated in number, you won't recover the amount of genetic variation that you had from the original population. So for example, you have northern elephant seals that underwent what's called a bottleneck in the 1800s. There was overhunting, and the number of northern elephant seals got very, very small. So you started from, the reason this is called a bottleneck is that you started from kind of a large population. The width of this is indicating a large population, and then in a small amount of time it narrowed to a small population. So this is the result of hunting, overhunting. And then they took that small population and the government protected the elephant seals, so they were able to reproduce with each other within the small group and recreate the large numbers again. Sorry, it's a little defective there. But what's interesting about this is even though you can recreate the size of the population through breeding programs, that new population, that repopulated group, this new population, will not have the same level of genetic diversity that the original population did. I'll call this old pop. So this is called a bottleneck because just in terms of numbers, you go from a large number of individuals to a small number and then back to a large number. They should probably call it the hourglass, but anyway, they call it the bottleneck because you have this time where the species is, fundamentally endangered. And you could argue that the new population is still endangered because they don't have enough genetic diversity to make them viable long term in the face of evolutionary pressures, selection pressures. Okay, so evolution is genetic change over time. That's the basic definition of evolution. And we've been talking about things that can cause genetic change over time in a population. High levels of new mutations, migration between genetically different populations, genetic drift in small populations. These are all things that cause change, genetic changes over time. But only natural selection is the force that produces adaptive change, meaning change that increases the fitness between an organism and its environment. Okay, so here you have a table. Of course, they show the panda bear. Most of the animals that you see in the Arctic will have the white fur because it helps with camouflage. Camouflage is a very important feature for survival, and so over time, animals have been selected, or white color of animals is selected favorably amongst a population. So over time, more of the organisms have the white coat color. You'll see rabbits, foxes, polar bears, they all have the white coat color. Anyway, so here in this figure, mutation, migration, genetic drift, and natural selection all change allelic frequencies, but only natural selection has, well, it doesn't say it here, but the ability to produce adaptive change, which increases the, it's only consistent at producing adaptive change. Mutation, you could get lucky and get something adaptive, but you could equally get something negative when you have a new mutation. Migration, again, could be positive or negative, but only natural selection will reliably give you adaptive change in the group.